rush this talk.
This is coming off your time.
Now, the joke is that at Ohio State, this sort of thing works fine.
Yeah. Thank you.
All right. So photosynthesis can be studied at many different scales, all the way from the leaf to the ecosystem, and these scales are all interconnected and they'll feed back on each other. So for example, the arrangement of leaves at the canopy scale is going to affect how light is distributed among the trees occupying that canopy. This is going to cause physiological and biochemical acclimation in those leaves, such as the amount of nitrogen invested in these leaves and its partitioning between chlorophyll and rubisco. But this light variability is also going to feed back on the canopy structure by causing branch growth in sunlit areas and branch depth in heavily shaded areas. And this differential branch depth and growth is therefore going to cause a change in the amount of the leaf distribution through time among the years. And presumably, this arrangement of leaves in time is going to determine how different canopy microhabitats vary from year to year. And this can have important effects on the morphological acclimation of the leaf, because that is largely based on the light reaching the bud from the previous season. So if the light around that bud environment the previous season is very different than the current year's light, you might have the leaf morphologically acclimated to the wrong kind of microhabitat. And so you can see how both the current and previous year lights both have an effect on leaf acclimation to some extent, and that's going to determine how the ecosystem functions at the upper scale as far as the gas exchange. And then focus of my talk today is really going to be sort of in this intermediate scale. And so the way we measure canopy structure in our group is we use a portable canopy lidar. And this is worn by an operator that walks at a constant pace along the forest floor, and it shoots up near-infrared pulses that bounce off canopy surfaces. And the machine records the amount of time it takes for lasers to return back to the system and how many lasers are returned. And this data is then binned into 1-meter columns along the axis walked by the operator, and it's further binned within those columns into 1-meter square bins based on the number of returns and how long it takes for them to come back. And so this essentially creates a two-dimensional matrix of the distribution of the canopy elements in horizontal and vertical space, and then this can be used to quantify the canopy in many different ways. The main metric we use is something we call ergosity, and this is the horizontal and vertical variability in leaf distribution. So you would take the standard deviation of the leaf distribution in each of the vertical columns along the axis, and then take the standard deviation of those values from each other. And higher ergosity values equal more leaf variability. And so over the years, what we found is that as forests increase in age, the ergosity tends to go up. This increase in ergosity increases the resource use efficiency and carbon sequestration of the forest, and has important effects on light distribution. Some other canopy metrics that we've been calculating are porosity, which is just the amount of empty space in the canopy. And this is calculated by adding up all of the empty bins with no returns up to the last return in each bin, and then dividing by the total area of bins that make up that area. We also have the top of the canopy, which is just the average of the last returns in each column. And then we have an average weighted leaf height, which is just the average height of where most of the leaf material is located. And so, for example, if we took a theoretical column of leaf returns at 1 meter, 3 meters, and 5 meters, the average height will obviously be at 3 meters. But the weighted height takes into account how many returns are at each level. So you would add up the returns at each level and divide by the total amount of returns. And so you can see the weighted leaf average is actually slightly lower because there's more hits at the 1 meter level, so it drags down that average. So for the talk today, I'm going to be focusing on these structural metrics, but we've come up with some other ones. Due to time constraints, this is all I have time to talk about. And so the idea here is that given our a priori knowledge of all the things that ergosity does, this is going to be the driver in this conceptual model that I presented. And the first objective is to see how do all of these other structural characteristics relate to ergosity. And then finally, how do all of these metrics, including ergosity itself, vary over time as a function of how ergose the plot is at the beginning of that time. And so my research is conducted up at the biostation in the permanent plots set up around the Meriflux footprint. And these plots have been lidared on and off since 2009. And I chose only plots that had no more than 1 year of missing data. So each plot had 5 to 6 years of lidar data for a total of 52 plots. And then I basically calculated a mean, a coefficient of variation, and a slope for each of those 4 metrics that I talked about earlier, where the CV is just the standard deviation of the metric across this time divided by the mean of the metric. And the slope is just how much that metric is changing over time. So if it's a positive slope, that metric is increasing in magnitude. And so the mean, CV, and slope of all the metrics are going to be my independent variables, and they're all going to be regressed against the mean ergosity for that plot to see how they vary over time as a function of ergosity. So for the first comparison I'm going to be looking at is ergosity against ergosity. So essentially each one of these points represents a plot, And the x-axis represents that plot's average rugosity during the census period, and the y-axis represents how much that rugosity is varied from year to year in that plot. And as you can see, there is a significant negative relationship, so plots that are more complex, have a higher variability of leaf distribution, tend to have less variability in that leaf distribution among years. And we look at the slope, where again, each point is a plot, but the y-axis this time is the change over time. So anything falling above that dotted red line is 0 means that the rugosity is increasing over time from 2009 to 14. Anything below the line means it's decreasing in time. And as you can see, there's not a significant relationship. So more complex canopies aren't becoming more or less complex over time. Next comparison will be the porosity, or again, the empty space in the canopy. And this is the spatial relationship. So this is just the average porosity over the census period rugosed against the average porosity over the time period against the average rugosity. And you can see more rugose canopies just have more empty space with no branches or leaves in it. The amount that the empty space varies between years is actually an inverse function. It decreases as the complexity of the canopy increases. So a more complex arrangement of leaves has less variability in how much empty space is in that canopy from year to year. When we look at the slope of change, you see there's no directional change. So more complex canopies are not becoming more empty or more open over time, or less open or empty over time as a function of how complex the canopy is. Next, we'll look at the canopy top, which is the average of the last returns. As you can see, complex canopies have a higher leaf distribution, or higher top of the canopy. That top canopy varies less among years as the rugosity of the plot increases. And again, there's no directional change, so complex canopies aren't becoming taller or shorter over time. And then finally, I want to look at the average weighted leaf height, which is the average height of where most of the leaf, or most of the canopy material is found. And as you can see, there's a higher average height of where most of the canopy stuff is found in higher rugosity canopies. This, however, does not, the variation of this average leaf height is not affected by how complex the canopy is. However, there is a directional change in where the maximum leaf area is distributed. So there seems to be a downward shift in leaf material over time as you move to more complex canopies. So I just took an example of one of these points. You can see that in 2009, the average height was 8.4 meters, and it almost eroded away 2 meters over the 6-year census. So there's a downward, you see the downward shift in the leaf material over time. And so just to sum all these up, all the structural metrics that I've looked at in this, at least in this presentation, all seem to have a positive relationship with rugosity at one instance in time. So more rugose or more complex canopies have more empty space, they have a taller canopy, and the leaf material is distributed higher up in the canopy. All of these variables, except for the weighted leaf height, seem to vary less between years or among years as a function of how complex the canopy is. And then finally, only the average weighted leaf height is changing in any kind of predictable way over time. So that's kind of an important thing. None of these variables are changing in a predictable way, which means presumably the light habitat is not changing in any kind of predictable way. So if the canopy was becoming more open, maybe you would see it, you would imagine there's a slight increase in light over the years, and you would expect the habitats to become more open and there'd be more light, but they're changing sort of in very unpredictable ways as a function of how complex leaf arrangement is in any point in time. And so what I'd like to explore after this is sort of what effects this might have on the actual leaves grown in those canopies. And so what I've started last year and I plan to do next year is I've been taking hemispherical photographs of different areas in the canopy by attaching a self-leveling mount to a pole and taking hemispherical photographs. And I've taken about 800 photos a summer. And I'm going to be taking these pictures in the same spots every summer and following these microhabitats through time and see how much they vary and see if they're changing in any kind of predictable way as a function of the arrangement of leaves in space. And then finally, this summer, I hope to implement a canopy manipulation experiment. I'm going to take Zeke up into the canopy. I'm going to try to shade different branches and shade them before the bud burst that gives rise to leaves, and shade it after its burst. Basically, manipulate the microhabitats and see how well the leaves are acclimated to the current year light and how much their acclimation was based on the previous year's light, because I'll be able to manipulate that with shading cloths. And with that, I'll take any questions.
Thanks. Do we have time for a comment or a question? Okay.
You mentioned there wasn't much change in the, you didn't see much change over the period of your study.
as far as like the, well,
Canopy height?
yeah, a lot, well, at least the variables that I've looked at, yeah.
Are there other studies that are looking at the same variables that have seen other ecosystems where they do have that? Or?
I'm not really aware of anyone who does this kind of work on this kind of temporal scale. So maybe people will LiDAR every 5 years and see how it changes, or they'll look at like seasonal changes in phenology, but not sort of year to year at this kind of spatial resolution. So I'm not aware of any studies that have done that. But the interesting thing, it seems to be going slow enough that you could perhaps measure it every 5 years, just to save money as far as like your sampling effort. But there might be ecologically important questions by sampling it at this kind of scale. If you look at the kind of the variability and stuff like that, that you couldn't get from a 5-year census. You can get like the slope of change, but I think this variability is going to play an ecologically important role, and that's what I would be exploring.
Okay, thanks, Alex. Cynthia Scheuermann?
Amy's going to go.
Oh, Amy's next.
Yep, sorry about that.
From Virginia Commonwealth University, graduate student working with Chris Goff.
Should just be able to speak. Okay.
Let's look at mine.
Okay, so I'm looking at factors that drive coarse woody debris respiration after a moderate forest disturbance. Okay. So a forest disturbance is just anything that results in tree mortality within the forest, and it's usually categorized into kind of two groups. So the first would be a severe disturbance, and these are usually catastrophic events that result in mortality in all trees or most of the trees. And so these are commonly caused by catastrophic wildfires, clear-cut harvests, or extreme wind throws. And moderate forest disturbances are disturbances in which only a subset of the forest trees have been killed. And these are characteristically caused by insect outbreaks, pathogen outbreaks, or rapid age-related senescence of certain forest trees. And so any type of forest disturbance that results in tree mortality alters the carbon balance of the forest. So it alters kind of the relationship between how much carbon is taken in by the forest through photosynthesis and how much is released back to the atmosphere through respiration. And so whether a forest shifts from a carbon sink, where it takes in more carbon from the atmosphere than it releases, to a carbon source, where it releases more CO2 to the atmosphere than it takes in, is largely dependent on the amount of coarse woody debris produced. So this takes into account the number of trees that were killed by the disturbance event, and also kind of the amount of carbon that's moving from being stored in living trees to being in a detrital pool. And then it also depends on the rate of coarse woody debris decomposition. So how fast is the organic carbon that's stored in woody debris being released back to the atmosphere? It could be released fairly quickly as kind of a pulse of CO2 back to the atmosphere, or it could be a slower rate of decomposition where the carbon is released at kind of a slower rate over a longer period of time. So my objective with this study was to look at different biotic and abiotic factors that could potentially drive coarse woody debris respiration, or respiration from the heterotrophs that are actually involved in the decomposition of coarse woody debris. So I looked at two physical factors, temperature and wood-water content, which have previously been shown to drive coarse woody debris respiration. And then I'm also examining the decomposer functional community composition. Okay, so I did my experiment in Facet. You'll remember that this is the ecosystem level manipulation where all early successional aspen and birch trees were stem girdled in 2008. And so this resulted in a pulse of 35 megagrams per hectare of carbon as coarse woody debris into the ecosystem within the disturbed area. And so I added to coarse woody debris syntheses, where I went out and looked at kind of the pools of carbon at different levels of decay within the ecosystem from 2009, 2011, 2013, and then in 2014. And then I also took repeated measurements of instantaneous CO2 efflux from coarse woody debris. And with each efflux measurement, I also took wood temperature and wood water content, volumetric wood moisture. And so, oops. So this is the method that I use to measure CO2 efflux. It's with the light core. You create a closed chamber. And so I did these measurements on 3 replicates per decay class. So a decay class is just kind of a group of coarse woody debris based on different characteristics. And it describes the level of decomposition. And on each replicate, I had 5 sample points that were evenly spaced along the length of down woody debris or the height of standing woody debris. And so this is a picture of Chris Vogel and I up measuring respiration from the top of a standing piece of woody debris. Okay, so the first thing I looked at was how coarse woody debris carbon stock changed over time. And so you can see here that in 2009, most of the trees that were stem girdled, in fact, it hadn't yet reached mortality. But in 2011, all stem girdled trees had been fully killed. So that's when you see. Quit. Okay. So in 2011, that's when you see kind of the first pulse of the dead wood entering the down woody debris categories. And then through time, you can see how they change as they progress through decomposition. And then this is the standing woody debris, and you'll notice that it's on a different axis, and this is because it by far makes up the largest stock of carbon within the ecosystem. And so just kind of a mass balance metric from 2011 to 2013, standing woody debris decreased by about 12 megagrams of carbon per hectare, and down woody debris only increased by about 9.5 megagrams of carbon per hectare. So we have a loss of almost 3 megagrams of carbon per hectare, either decomposed and released to the atmosphere or retained on site as soil organic matter. Okay, and then I also looked at instantaneous coarse woody debris respiration rates over the summer, and these were really variable within decay classes, among decay classes, and over time. But one important thing that we have noticed from this is that standing woody debris, which has often been thought to decompose extremely slowly and respire at a really slow rate, doesn't respire at a significantly lower rate than decay class 1 of down woody debris. So this is kind of an interesting result. And so then if you look at it from an ecosystem perspective, after a moderate disturbance, standing dead wood in the system could become a more significant source of carbon flux out of the ecosystem because it comprises such a large portion of the carbon stock in the ecosystem, and it respires at a relatively high rate compared to what many people have previously thought about it. And so what this figure is showing is NEP and ecosystem respiration for 2013 that were measured from the flux tower. And then these are the the components of ecosystem respiration. That's an estimate of how much coarse woody debris from each decay class might contribute. It's modeled, so it might contribute to ecosystem respiration. And you can see this big black bar is from the standing woody debris. So it could be a pretty significant contribution. Okay, and then what I'm currently doing is I'm looking at extracellular enzyme activity within the wood. So I'm looking at 4 fungal enzymes that are known to be involved in wood decomposition. So lactase and manganese peroxidase are involved in the breakdown of lignin, and beta-glucosidase and beta-glucanase are involved in the breakdown of cellulose and hemicellulose. And so what I'm really trying to get at here is how enzyme composition changes with the extent of wood decay. And so this is what I'm doing right now. So I don't actually have results from this, but stay tuned and hopefully I'll have some. Great. Any questions? Yes.
Is there any chance that the respiration of standing dead wood is in fact quite low, but that it's channeling respiration from the portions of the tree that's in contact with the ground?
It could be, but we took different measurements at different heights, and so it wasn't extremely variable between different points along the tree, but that could definitely be where the CO2 was coming from. So it's not that it's like a chimney effect all coming out of the top, sustained along the height of the tree. Yes.
Well, one way to kind of get at that question Luke had is what kind of state, what decay class are these standing trees in? So you have decay 1, which is essentially a brand new fall down tree, just died. Decay class 5 is getting ready to melt into the soil. So where would you characterize these standing trees?
Oh, the standing dead trees, we kind of characterize them as being similar to decay class 1.
So there hasn't been a lot of composition of those trees yet?
Not yet.
Hey, thank you, Amy, and thanks a lot.
Mm-hmm.
So our next speaker is Cynthia Scheuermann from Virginia Commonwealth University.
Where did it go? Where is it? Oh, there it is, right there. Sorry. Is it gonna work? Yep. Just gotta speak up. This is the laser, this is the advanced.
Okay, thanks. Is that me? Okay. My name is Cynthia Scheuermann. I'm a first-year master's student from Virginia Commonwealth, and I'm going to be talking about my research project that I'll be completing the research for this summer at the biostation. The title is Forest Physical Structure as a Proxy for Net Primary Production over Decade to Century Timescales. Didn't work. Okay. So I'm really focused on the carbon aspect of forests, the potential for forests to sequester atmospheric carbon, and the part of the equation I'm going to be looking most at is net primary production, basically the rate of forest growth. So in the eastern United States, we have forests progressing along successional age, though the mechanisms that are involved in carbon sequestration over these advancing timescales are less understood, so that's what I'm going to be looking at. So this is some previous work that I've done that's comparing synthesis of papers reporting net ecosystem production over 3 countries or 3 continents and comparing them to forest age. If you can, it might be too light to see the gray box, but the time period between about 75 years of age and 120 or so years of age is sort of the middle successional time period I'm interested in here. But the results of this are that forests are not declining in NEP as they age, or there appears to be no negative trend, which means that we need to understand the mechanisms that are driving these carbon trends over successional time. So I'll be looking at forest physical structure, which I'm going to define here as the arrangement of leaves and stems within the canopy. Ecosystem structure and ecosystem function are closely related. And as above ground physical structure changes when forests advance in age and composition, it will influence the function of the ecosystem as well by changing the distribution of resources such as light and nitrogen that support growth. So structural metrics I'll be looking at will be leaf area index, gap fraction, rugosity, and other LiDAR products that Alex mentioned previously, and that'll be one of the systems I'll be using. Okay, so my first objective in this research is to use multiple imaging approaches to determine how forest physical structure varies over ecosystem development. And with disturbance frequency, I'll be working in the LTREB plot, so I'll actually have two parallel chronosequences of different disturbance frequencies, well, disturbance intensities. So this is a theoretical trend of forest complexity over successional time. And one of the hypotheses I'll be following is that forest stands that are more frequently disturbed may lag in structural development over this time period. So I'm going to be using multiple imaging techniques, and these are going to be increasing in measured dimensions. I'll use these to identify which expressions of canopy physical structure best serve as proxies for NPP. And my hypothesis here is that the more measured dimensions you get, so as you move into 2D and 3D measurements of forest complexity, you'll get better predictors of net primary production. My third objective is to link forest physical structure with canopy light interception as FPAR across ecosystem development, and this has to do with the distribution of resources that will be changing as forests age. I may also be looking at nitrogen as well, which is going to be another resource of interest. So I'll be using the LTREB plots, and this is an 11-stand chronosequence. And these are gradients of age and disturbance severity. So we have, since initial burning and clear cutting of the region around the biostation, we've experimentally cut and burned a series of burn and clear cut plots and a series of clear cut only plots. I'll be comparing these on the LTREB. The reason using a chronosequence approach here is useful is that because the changes in forest physical structure, these are forests that have shared climate and shared soils, generally speaking. So it'll be ideal for trying to figure out the changes in structure that are impacting the changes in MPP. Okay. So these are the three imaging and LiDAR techniques I'll be using, ranging from more traditional to more exploratory, using hemispherical imaging, a 2D portable canopy LiDAR system, and a 3D ground LiDAR. Hemispherical imaging is a one-dimensional measurement, basically a 180-degree view of the canopy, and I can analyze these images for metrics such as canopy openness, gap fraction, and leaf area index estimate. The 2D portable canopy LiDAR allows you to see the development of layers in the canopy itself. You'll be looking in horizontal and vertical dimensions. And metrics I'll be getting from this include rugosity, canopy gaps, canopy openness. A 3D small footprint LiDAR is a method I used last summer to get point clouds of LiDAR returns in each of these stands. And this is basically a unit that will go in a 360-degree rotation and produce a point cloud of vegetation in that plot. So these will be combined into per plot, and then we're working on image processing for structural metrics with some partners at Rochester. Other measurements of interest are FPAR measurements, which will be useful in determining how resources are being distributed over these forests that differ in age. Also, NPP measurements will be collected as part of the larger LTREB project. And this is a sum of wood NPP and leaf NPP. So the reason I'm doing this project is to link the measured proxies of NPP and FPAR in order to improve understandings of the structure-functional linkages of carbon over successional time. and seeing which proxies or which structural metrics are more useful proxies at certain periods in succession might be useful for finding mechanisms, or the mechanisms that are actually happening over these timescales. This will also be a useful comparison of multiple imaging and LiDAR methods for anybody looking to measure forest structure with any of these methods. Any questions on this?
Thank you, Cynthia. Comments or questions?
I have a question.
Yeah.
Can you geolocate the stems from that ground-based LiDAR or from the 3D?
We should be able to. We have the GPS coordinates of each of the scans we've taken. You can find where the stems are in the plot, so should be working on it, but that is a great idea. I'll work on making that part of it.
All right.
Here's your pointer.
Thank you. All right. How's it going?
Okay, our next speaker is Holly Andrews, graduate student in Mark Hunter's lab at the University of Michigan. While Holly's miking up, I should let you know that she represents biological stations at the Association of Ecosystem Research Centers in October. And there's a nice link we can send you to, she journaled her experience going to congressional offices, as well as the AERC meeting. And I understand you're blogging on that.
Yeah, there's a blog up as well.
Okay, so thanks for representing us. And now we're going to hear about some of our research, so thank you.
Okay. Can I just use the keyboard this time? Okay, thanks. Okay, so today I'm going to be talking about my master's thesis research. I'm Holly, master's student in Michigan's EUB department, and I conducted my master's thesis research at UMBS, which I'll talk about today. So there's a lot of evidence that climate change is currently occurring in our world today, and while most people focus on the changes in temperature, I believe that equally as important are the changes in precipitation that we're seeing. So we're expecting that our global average precipitation is going to increase 1 to 3% for every degree Celsius of temperature increase we're seeing. However, we know that precipitation is different across regions, and so we're expecting these changes to be different across regions and latitudes. In addition to average changes in precipitation we're also going to see increases in intensity and frequency of extreme weather events, such as flooding and drought. So how will plants be affected by this? We know that plants have evolved multiple mechanisms to deal with water stress, herbivory, all that good stuff. So if precipitation is changing, we would expect that those mechanisms would also change as well, and across different latitudes. And if plant defenses are changing, we would expect that to feed back into herbivore behavior as well as herbivore performance. So for my thesis, my overarching questions were, how will changes in mean precipitation and variation around that mean affect plant growth and defense across latitudes? And how will changes in precipitation indirectly affect herbivore growth, defense, and behavior? For my study system, I used the milkweed study system, a great plant to study because it has multiple defenses it uses against herbivory, and it's a native species to a lot of the US. So I collected seeds from 1 to 3 latitudes per plant species in the eastern USA, grew them in a greenhouse, and then I also did a subsequent study where I used monarch caterpillars, which are specialists on milkweeds, and they actually are able to sequester some of the defensive toxins that milkweeds produce, called cardenolides, and use them in their own defenses against their own parasites. So I conducted 3 experiments to test these questions, and I'm only going to talk about 2 today. One was a common garden experiment that I did at the UMBS greenhouse to test effects of precipitation on growth and defense of milkweeds from multiple latitudes in the eastern US. And the second was an experiment I also did in the greenhouse to test indirect effects of precipitation on growth and defense of monarch caterpillars on one population of one species of milkweed. Okay. So for my first very large experiment, I used four species of milkweeds that sort of differed in their habitat preferences. Some preferred wetter habitats, drier or intermediate. And then for each of those species, collected 3 latitudes, actually for 2 of those species, except for Asclepias viridis, which prefers drier habitats, which I collected from 2 latitudes, and Asclepias quersavica, which I collected 1 latitude. And then for each latitude, I collected 3 populations for replication. Okay, this is going to get a little complicated, but for each of these populations, I applied 6 different precipitation treatments. 3 where I changed the mean precipitation, so they received the same amount of water every day, but those levels were different depending on my wet control or dry treatments. I also did 3 precipitation treatments where I changed the variation in how much water they received per day. So when I talk in my results about changes in variation, I'm talking about any of these versus any of these. And you can see that they're wet, control, and dry. Yeah. Okay, and for my second experiment, I used 1 population of Asclepias syriaca, which is sort of an intermediate wet preference species, applied those 6 water treatments, allowed the plants to grow for about a month, placed first instar caterpillars on 1 caterpillar per plant, allowed them to grow for 5 days, voided their guts for 24 hours, and then weighed the caterpillars for mass gain as a proxy for growth, and cardenolide content, which is the toxin, if you remember, for a proxy of defense. And these are all the characters I tested throughout my thesis, but I'm only going to talk today about 2 milkweed defenses and both of the herbivore performance traits. So what did I find? First, let's look at latex production. So milkweeds are called milkweeds because they produce this very white, milky, latex-filled sap. And those can serve as a defense against herbivores. To orient you to this figure, the color scheme that you see here applies to latitude. So high latitudes are in white, low latitudes are in black. And then latex production on the y-axis. And for Asclepias incarnata and syriaca, I found significant differences in variance in precipitation. So whether there was any variability in precipitation or not. And so for Asclepias incarnata, we found that high latitudes produce less latex. However, variation in that precipitation caused a decline in latex production at mid-latitudes. And if you look at the bottom now, for Asclepias syriaca, high latitude populations again produced less latex, but variation had a negligible impact on production. Okay. For Asclepias viridis, remember I said I only collected from 2 latitudes, so you see the same color scheme here, but only high and mid latitudes are represented. And variation in precipitation eliminated the latitudinal variation that we see in latex production. So when there was no variance in production, you can see there's quite a difference that goes away when we add in variation. Okay. So in summary, I didn't see any effect of changes in average precipitation on latex production. However, increasing variance in precipitation caused declines in latex production at mid-latitude for Asclepias incarnata and high latitude for viridis. And low latex is known to increase the susceptibility of milkweeds to caterpillar damage, so we might expect adverse effects in the future if our latex production is decreasing. For cardenolides in our plants, first of all, looking at overall cardenolide content in each of our species, we can see that there are vast differences in how different species allocate carbon for these defenses. And so we can see that Asclepias quersavica produces the most cardenolide, while Asclepias incarnata produces the least. And here we did see differences in mean precipitation. For incarnata, only high latitude populations produce any cardenolide at all, which is quite fascinating. And for Asclepias syriaca, high and mid-latitude populations produce more cardenolide under our drier conditions. Here on the left, let me not fall over, while low-latitude populations produce more cardenolide under wetter conditions on the right. And for Viridis, again, remember we only had 2 latitudes, and variation in precipitation caused the decrease in cardenolide production for our high latitude populations in white. Sorry, I'm a little sick. So in summary, milkweed species we know differ in their allocation of resources, and cardenolides were only produced in high latitude populations of Incarnata, while changes in average precipitation for Syrica and variance in precipitation for Viridis have differential effects on cardenolide production depending on latitude. And this could be some sort of genotype by precipitation interaction if we assume that our milkweeds are of different genotypes, which could affect evolution, could have evolutionary consequences. Okay, I'm going to fly through what I found for my caterpillars real quick. So when we looked at our caterpillar growth rates, we found that caterpillars actually grew more slowly when they were reared on wetter plants. Remember, this is only one population of Syrica being used, but there might actually be some reason for that. So I went back and looked at that one population of Syrica that I used and assessed its own cardenolides for just the one population. And we see that Syrica actually produces more cardenolides, so that could be what's causing lower growth rate in our caterpillars. And for cardenolide sequestration in our caterpillars, we see a difference in not mean precipitation, but variation in precipitation. They sequestered more cardenolides when reared on Syrica exposed to increased variance in precipitation. And we can't really find a reason for this yet, but it might not be directly related to plant cardenolides, but rather something about changing nutrient status. Okay, so in summary, an increase in average precipitation caused our caterpillar growth rates to slow, and an increase in variance in precipitation caused increased cardenolide sequestration. I think this picture is adorable, by the way. So while I haven't analyzed all of my data yet, I wanted to present you with some conclusions so far that plants at different latitudes have different strategies for defending against herbivores, and these strategies may shift as precipitation changes. And as plant defense strategies shift, this could have consequences for herbivore growth and defense. And thinking even higher, there could be impacts for other trophic levels, such as this predator, this crab spider eating a caterpillar. So yeah, I want to acknowledge everybody, and especially the biostation, for housing me and providing some funding for me. Any questions?
Thank you. We have time for a comment or question while Claudia mics up. Comments or questions for Holly?
It was that awesome.